Hello, welcome back, everybody, to Virology. Today we're going to, this is our last part of this course where we're going to talk about things that go on in individual cells. We're going to talk about, the rest of the course we're going to talk about infections of hosts, which is probably to you the most interesting thing, because there's no formula for MOI, stuff like that. But before we do that, I want to have an overview of what we've just talked about. I call this the infected cell. I want to talk about how the cell and the virus infections interplay with each other. So we have so far focused, as I said, on viral gene expression, genome replication, assembly of virus particles in cells and culture. And as you know, they depend on the cell, because that's why we call viruses obligate intracellular parasites. They depend on cell functions. And they depend on metabolic functions, which we haven't talked about too much, biosynthetic functions, signaling, and trafficking systems. Trafficking we've talked a little bit about in terms of microtubule transport and so forth. But today we're going to talk about metabolic and biosynthetic and signaling pathways. So what I want to do is give you a description of the impact of virus infection on a host cell. You already know viruses get in, virus particles get in, they reprogram the cell to make more viruses. And today we're going to see the effect of that. This just reminds me, Stan Falco was a microbiologist, really good guy who studied microbial pathogenesis. And he used to say, the goal of a bacterium is to make another bacterium. Now, bacteria don't have goals, just like viruses don't have goals, but you get the picture. So it's the same with viruses. The only thing a virus needs to do is to make more viruses. Everything else is incidental. Remember that. So when you read a headline saying the virus wants to become less virulent or more virulent or spread to more hosts, this is pure BS, all right? Because there is no desire. It is all driven by making more virus particles. And if you get one thing out of the course, remember that. A virus needs to make more viruses. That is it. It doesn't want to make you happy. It doesn't want to make you sad. It doesn't want to make you healthy or sick. It doesn't want to do anything. Unlike you who have desires, viruses have none. That's a good quote, isn't it? Yeah, I like that. So we're going to talk about today 4 things. We're going to talk about signal transduction. We're going to talk about gene expression. We're going to talk about metabolism and remodeling of cellular organelles. These are things we really haven't talked about much so far. So signal transduction first. You know what this is. You find it very boring. To memorize pathways where something binds something, there's a phosphorylation cascade that binds something else. Yeah, it's really boring, because I think the most interesting part is the consequence, right? So I'm going to show you pathways, but you don't need to know them. I'll show them to you to give you a sense of what I'm talking about. So as you know, a cell needs to respond to its environment. It needs to know if there's food around or if not, and other things as well, temperatures, pH, and so forth. And that's what signal transduction pathways do. They do everything in the cell. They govern every kind of cellular activity. And virus infections can lead to modifications of these signaling pathways so that the virus reproduces better, so it makes more viruses, right? And so here's an example of a signaling pathway. This is a pathway, it's called the PI3K AKT mTOR signaling pathway. I would never ask you what that stands for, okay? It's just to know that this is what the pathway is. What this does is, normally in the cell, various ligands bind to receptors at the cell surface. For example, growth factors. So if there's a growth factor present, the cell will divide. And how does it do that? Well, through signaling various phosphorylation events and turning up, say, cell growth and proliferation, cell division, and so forth. And what you see superimposed on this are green and red arrows, where viruses either stimulate that part of the pathway, that's green, or inhibit it, that's red. So here, Here, stimulate or inhibit. There's the key at the bottom. So herpes and viruses here, papillomavirus, inhibit this protein, this TSC2-1 complex. Now you have to be really careful when you interpret these, because that inhibition actually has a positive effect, because this protein normally inhibits Reb, so the rest of the pathway doesn't happen. But when adenovirus inhibits TSC2-1, now Reb is active and you get activation downstream and more proliferation in cell growth. And of course, that's beneficial for the virus to have more cell reproduction. So, you have to be a little subtle when you interpret the effects of these arrows. But the simpler ones are here at the top, where binding of ligands to these plasma membrane receptors will cause a positive effect on PI3 kinase. So PI3K is a kinase that will then phosphorylate other proteins. Okay, so for example, in entry, when viruses bind receptors, the binding is not simply to take the virus particle into the cell. That's an important part of it, but it leads to other molecular events. And there are three of them shown here for illustration. So on the left, we have adenovirus binding two different receptors. Remember, there's a fiber receptor, and then there's an integrin receptor, which leads to internalization. The signaling, the binding to the integrin receptor sends signals into the cell. It's almost as if the ligand for that receptor were present. These are these green arrows here. So the binding stimulates endocytosis. This is an upregulatable pathway, the endocytic pathway. It also stimulates the activity of these proteins, Cas, CSARC, PI3K, which activate other proteins, which in turn depolymerize the actin so that the endosomes can move into the cell, right? When we see movies of endocytosis, it looks like the virus binds and the vesicle moves in the cell, but lots of things are happening. There's an actin microfilament layer beneath the plasma membrane, not even shown on these pictures. It's just a little bit here. That has to be loosened up in order for the endosomes to come in. So binding to the receptor participates in the loosening of the actin microfilament. In the middle one, we have influenza virus doing a variety of things to facilitate entry. Let's see so you can see the binding is causing other proteins on the surface to be activated. These are receptor protein tyrosine kinases. Normally they're present by themselves as monomers. When they're activated, they dimerize and become active, and they can phosphorylate other proteins. So what they're doing is activating a pathway here through PI3K that again is loosening up the actin microfilaments. This signaling also leads to the stimulation of entry of protons into the endosome. So that's part of the normal endocytic pathway, that binding stimulates proton uptake to lower the pH of the endosome. And then there's another one here where virus binding to sialic acid alone stimulates calcium channels. Activity of, these are voltage-dependent calcium channels, VDCC, and calcium comes in, and that facilitates the exit of the RNPs from the particle. So lots of signaling activities going on. And finally, we have Ebola virus binding to cells, being taken up, as you can see here, by this surfing mechanism, ruffling, and the activation of the receptors leads to a PI3K-dependent pathway, which actually blocks, this is the Racaniello protein here, you see, RAC. Actually not, but it's cool. It blocks the movement of the endosome to a vesicle that would be non-permissive for fusion. So the virus particles will exit the endosome here. If they went further, they wouldn't be able to get out of the endosome, and so this signaling actually facilitates their infection. Okay, so that's three examples. And there are many other ways that virus infection activates signaling pathways, not just through binding and entry. Here are two intracellular pathways. Again, it's the PI3 kinase mTOR relay. mTOR is a major regulator of cell metabolism. And here you can see that mTOR can upregulate. mTOR is a protein complex. It can upregulate translation. And of course, more translation is good for viruses, right? Make more proteins. So you can see here, these are viral proteins, hepatitis C virus, NS5A, and NSP1 of rotavirus, E4-ORF1 of adenovirus. Doesn't matter. Point is they're activating AKT and leading to increased translation. And at the same time, they're inhibiting apoptosis, right? That's programmed cell death. You don't want the cell to die before you are finished reproducing. So that has a multiple function there. Same thing with hepatitis B virus, X protein, and this interesting protein from human herpes virus 8. It's a G protein coupled receptor, 7 transmembrane segments, activating again AKT to increase translation and inhibit apoptosis. Okay, so many ways that signaling can facilitate virus infection. Here's an interesting example of a viral RNA that is involved in signaling. So this happens with flaviviruses. These are these envelope viruses with plus-stranded RNA genomes. The genome is shown at the left here. It is not to scale, so the genome is much longer, as shown at the top there, encodes for a long polyprotein. But here's a schematic of the genome, the 5' untranslated region, the open reading frame, which would be the majority of the RNA. And then there's a 3' untranslated region, and both of these are highly structured. During replication, there is an exonuclease called XRN1. This will come back, so remember that, which an exonuclease, it chews from the end and degrades the RNA. So this exonuclease is chopping up the RNA through the ORF, and it gets all the way through most of the RNA. Remember, the 3' UTR is a very small part of the RNA. That's shown here. And then it can't go anymore because this secondary structure blocks its chewing. That RNA is called sfRNA. What does SF stand for? Small
Flaviviral.
Flaviviral. Thank you very much, because you work on it, right? Yeah. Very good. So you don't have to remember all this crap. Just names. Small flaviviral RNA. You need it for pathogenicity, and you need it for plaque formation because it promotes apoptosis. So it inactivates AKT. So remember, AKT here can regulate apoptosis. So normally AKT inhibits apoptosis. So if you inactivate AKT, you're going to get apoptosis, right? So the little RNA, the sfRNA, inactivates AKT. You promote apoptosis. That makes you make nice plaques, and it causes pathogenesis in an animal model. If you delete a little bit of this 3' end, you now allow the exonuclease, the XRN1, to chew the whole thing up. So there's now no sfRNA present, and that mutant does not make plaques. Deletion near the 3' end prevents production of the small genomic RNA, short genomic RNA. So this is essential for pathogenicity. So it's activating as a modifier of a signaling pathway. So you don't have to be a protein. Okay, so that's signaling. Let's move on to gene expression and what virus infection does to gene expression. And here is a diagram of gene expression, starting with DNA. This is cellular gene expression, right? So we have cellular DNA in the nucleus. We have transcription by Pol II, capping and polyadenylation and splicing, and RNA export and translation. Viruses interfere at every step. So here in red are the ways that viruses interfere with transcription. They can interfere with polyadenylation, with splicing, with RNA export. And once in the cytoplasm, they can interfere with protein synthesis, and they can lead to degradation of cellular mRNAs. So why do we have all of this interference with cellular gene expression? To reduce competition. So viral mRNAs have all the cellular translation machinery to themselves. There's no competition for cellular mRNA with bicellular mRNA. So you inhibit it at multiple steps, and you reduce the amount of cellular mRNAs that are made. So here's an example of inhibition of pre-mRNA processing. So splicing of cellular mRNAs by viral proteins. So we're now in the nucleus here in gray. Here's the cytoplasm at the bottom with a couple of nuclear pores. And you remember that splicing marks an mRNA for export. So that's what all these proteins that are bound to the pre-mRNA, some of them remain there after splicing, and that marks them for export. So that components of the nuclear export machinery like NFX1 can recognize these splicing proteins and export. So here we see that a protein of herpes simplex virus, ICP27, actually inhibits splicing. Then we have multiple proteins, multiple viral proteins inhibiting export of the spliced mRNAs through the nuclear pore. We have VSV proteins, we have influenza virus proteins. Influenza NS1 also inhibits polyadenylation. We have polio and rhinovirus proteins actually cleaving nuclear proteins that are responsible for export, like these NUPs, NUP153 and 62. They're actually cleaved, and also adenoviral proteins inhibiting export as well. So multiple points of inhibition of cellular export and processing by viral proteins. Now, normally in a cell, mRNA doesn't last forever. Every mRNA has a slightly different half-life, because some mRNAs you don't need for very long, and others you need for many hours or even days. And there are sequences in the mRNA that control degradation. And degradation is carried out by a series of nucleases in the cell. So here is a pathway for mRNA turnover. Well, we have an mRNA at the top there, and we have here a protein called CC4-NOT-PAN, all of which stands for something, in case you want to know what these letters are coming from. They stand for something, but I'm not going to tell you what they stand for because it's not important. This one chews the 3' end. It chews off the poly-A tail. PAN, poly-A nuclease. Okay, it's chewing off the poly-A nuclease. And then we have decapping enzymes that take the cap off of the RNA. And once the poly-A and the cap are removed, then other nucleases will chew up the RNA from the 3' end, so that's 3 to 5' decay, or our friend XRN1, which we already met, from the 5' end. So this is a normal cellular degradation pathway, which takes out mRNAs, depending on whether they have signals in them or not. And these proteins obviously would attack viral mRNAs if they weren't regulated. So in many virus-infected cells, some of these proteins are either removed or relocalized. And in poliovirus-infected cells, PAN, DCP1A, the decapping enzyme, and XRN1, they're actually degraded, so they can't be degrading viral RNA. So viruses need to get around this degradation pathway. And viral proteins themselves can lead to the degradation of cell mRNAs independent of these cellular pathways. So here we have mRNAs being translated. These are capped polyadenylated mRNAs. So here's a 5' cap, M7G, PPPNP. Here's a ribosome translating the RNA. And we have a couple of viral proteins here. We have a SARS-CoV protein, a HHV8 protein, a herpes simplex virus protein that are targeting different parts. The NSP1 and the SOCs are chopping up the RNA, as you can see here. And once the RNA is broken, once the cap is off, then XRN1 will recognize it as defective and chew it up. And also a vaccinia virus protein here leads to the same chopping up as well. So viral proteins can come and get rid of cellular mRNAs so that there's less competition, the same as inhibiting gene expression. You may ask, how do the virus RNAs avoid this? And they have specific sequences that are present in the RNA that make them resistant to this kind of degradation. All right, question 1. Which of the following is a consequence of viral proteins modifying signal transduction pathways to promote replication? A, polio inhibition of transcription by RNA Pol2. B, herpes simplex virus protein blocking pre-mRNA splicing. C, disruption of actin filaments to allow endocytosis. And D, initiation of mRNA degradation by viral proteins, which is a consequence of viral proteins modifying signal transduction pathways. Let's have a look at what we did. Most of you got C, which is correct. Disruption of the filaments to allow endocytosis. That's a signaling pathway. The inhibition of transcription by Pol II is by cleavage of a protein. Let's go back and look at that. That would be here. Polio infection inhibits Pol 1, 2, and 3 by cleaving them. Herpes simplex, protein blocking and pre-mRNA splicing. Where is that? Here, herpes simplex. And these are just direct acting proteins. There's no signaling involved there. And inhibition of mRNA degradation. Again, that is initiation of mRNA degradation. That is here. And of course, we looked at a lot of it here. This is just by endonucleases and exonucleases directly acting on viral RNA or cellular RNA. It's not a signaling pathway. Now, all of these viruses in our Baltimore scheme have a central common entity, and that's mRNA, right? They all have to make, in their reproduction cycles, mRNA that can be translated by the host cell. So now what I would like to do is focus on that translation step and talk a little bit about how viruses cause selective translation of their messages over cellular messages. So here's an overview of translation, which you all know cold, I'm sure. Yeah, it's okay. You're going to learn it now. Here's an mRNA at the top. It has a cap at the 5' end. And there's an AUG, an initiation codon, where protein synthesis begins. And the way the ribosome gets there is shown in this slide. The 40S subunit, remember, ribosomes, eukaryotic ribosomes have 40S and 60S subunits. So the 40S subunit first binds. It's brought to the 5' end by virtue of a bunch of proteins binding the cap structure. So here, EIF4E binds the cap, and then it brings in all these other proteins, including the 40S subunit. And then, once this is assembled, it begins to move along the RNA, and then eventually when the 40S subunit reaches the AUG codon, the tRNA that's included in this complex, that's the MET initiator tRNA, it recognizes the AUG, and a lot of proteins are released, and then the 60S subunit comes in. And then you can start adding amino acids to the next site on the ribosome. So that's how cellular mRNAs initiate protein synthesis, and many viral RNAs as well. But there are some interesting exceptions. Here's a series of cells infected with poliovirus. And you can see in the graph, we're looking at the rate of protein synthesis with the time post-infection. And the poliovirus infected cells, you can see by about 2 hours, the rate of protein synthesis is very much declined. And then it goes up again because it's replaced by viral protein synthesis. So cellular translation goes down, and then it's replaced by viral translation. And on the right is a protein gel, which illustrates that in a different way. Here we have time post-infection. We've labeled proteins in the cell with radioactive methionine. And you can see that at time 0, there's a lot of cellular proteins being made. But then at 1, 3 hours, you can see those cell proteins go down and down at 5, and by 7, there's no cell protein synthesis. It's all replaced by these labeled proteins, which are viral proteins. So this is an example of how a virus can lead to the inhibition of cellular protein synthesis and still have its own mRNAs translated. So how does that work? It works via modulation of the 5' cap. So again, I showed you in that original slide, the ribosome, the 40S ribosome, is brought to the RNA by interaction with a number of proteins, ending with interaction of EIF4E with the cap. And so this step is interfered with by virus proteins that's shown at the top. So for example, at the very top, we see that there's a protease encoded in some viral genomes that cleaves this red protein, EIF4G, so that now if EIF4E binds to a cellular cap, it can't recruit the rest of the proteins, including the 40S ribosome. So that effectively shuts off translation. There's some other modifications that happen here. This one is very interesting. There's actually a protein in the cell that regulates the activity of EIF4E. It is called 4E-BP1. When 4E-BP1 is bound to EIF4E, 4E can no longer bind EIF4G, so that inhibits translation. And so normally in the cell, 4E-BP1 is phosphorylated, which inhibits its activity. It is dephosphorylated in cells infected with polio and other viruses to activate it and shut down translation. So there are many ways that virus infection leads to inhibition of cell translation. Now, how does the virus get its own mRNAs translated, right? If it's shutting off the cell, it must be doing something different. So let me illustrate that for you. Here are two examples. So at the top again is your cellular mRNA translation, where translation of the mRNA requires all the eukaryotic initiation factors, these proteins that are bound to the 40S ribosome and to EIF4E. They're all eukaryotic initiation factors. There are many of them. And cellular mRNAs require the whole slew of them. Poliovirus and other viruses require most of them, except they don't need EIF4E because they don't have caps on their mRNAs, as you will see. And hepatitis C virus also only needs 2 of these initiation factors to start translation. So these viruses can trash cellular proteins and not worry because they can do it without them. So how does this work mechanistically? So here's an illustration of the poliovirus genome. This is the very first 800 or so bases of the RNA, which is shown here at the bottom. It's a plus-strand RNA, remember. And the very 5' end of the RNA is highly structured, which is shown without a translation at the top. This RNA, of course, encodes a long polyprotein, which we've seen before, that gets processed by proteases to make the final protein products. So there's a 740 base untranslated region before the initiating AUG codon. This RNA is not capped. It has a little protein linked to it. Remember that protein, VPG, was the primer for RNA synthesis. So there's no cap. So the virus can trash EIF4G because it doesn't need it. There's no cap on the RNA. But what does it do to get the ribosome to the RNA? Well, poliovirus and many other viruses have what we call internal ribosome entry sites, or irises. Think of the flower, the iris. It's different spelling, but sounds the same. So here are some irises from different viruses. You can see they're all differently structured. And in fact, there's no sequence homology among them. They have various structures that attract the translation apparatus. So for example, this one, this type 2 iris, a part of the iris here, which is highly structured, as you can see, the part in red, it binds EIF4G. So remember, EIF4G is normally brought to the cellular RNA by interaction with the cap binding protein and EIF4G. These viruses don't have caps. Instead, the 4G binds directly to the iris. And similar things happen for these other irises as well. Okay, so that is how these viruses, get around inactivating EIF4E. They don't need it because they don't have a cap. Rather, the RNA is directly interacting with the ribosome. And so you can see in this picture here, the internal initiation via an iris. The EIF4G is interacting with the highly structured region, and that can bring in the 40S subunit independent of having EIF4E. There's another way that translation initiation is regulated, and that also involves an early step in protein synthesis, and that's shown here. And this is interesting because this is now a cellular response to virus infection. So let me explain. Here we have the initiation step of protein synthesis, where we have our 40S subunit and the initiator met tRNA already bound. So this is going to eventually associate with a 60S subunit. And once the initiation codon is found, GTP, which is bound to the initiator tRNA, is hydrolyzed and it's released as GDP. That GDP EIF2 has to be recycled so it can be used for more MET initiator tRNAs. It can be added as GTP to new MET initiator tRNAs, and you get multiple rounds of translation. So how that happens is that the EIF2 portion and GDP are recognized by another protein called EIF2B, which acts as a guanine exchange factor. It swaps out GDP for GTP, thus recharging the EIF2 so it can rejoin the tRNA. So this cycle is very important for initiation of protein synthesis, this recycling of GDP for GTP. It's regulated. It's highly regulated in cells. And in particular, a couple of conditions will activate its regulation, and they're shown here. Endoplasmic reticulum stress, amino acid deprivation, and double-stranded RNA. So let's focus on double-stranded RNA, which you find in virus-infected cells. It's detected by a protein called PKR, which is a kinase. It phosphorylates EIF2 alpha, and that's the blue protein here. So EIF2 is normally bound to GDP and participates in the recycling reaction. But when a virus infects a cell, it activates PKR, and then EIF2 is phosphorylated. You can see in this pathway down here. It's got a little P on it. Now, when the GDP is released from the tRNA and EIF2 goes to bind it, it's locked in because of the phosphate. It can't be the GDP cannot be substituted for GTP because that phosphate locks it into the EIF2B. And the consequence is because EIF2B is limiting in the cell, eventually you have no more EIF2B because it's all bound up by GDP EIF2 phosphate, and protein synthesis is inhibited. So this is a way that the cell tries to inhibit a virus infection by turning off translation. That's not, in general, a really good thing to do, right? Turn off translation in a cell. And in fact, phosphorylation of EIF2 also leads to apoptosis, programmed cell death. But the idea is maybe you sacrifice those infected cells to save the organism. So you're going to reduce virus production, okay? So this is a really powerful system. So how can we still have viruses around? Well, because viruses counter it, of course. It's always the case. So here is an example. This is an example of how PKR, that kinase, is activated by double-stranded RNA. So here we have two molecules of PKR. These are made in the cell, and they are inactive as kinases. They cannot phosphorylate anything unless they bind to double-stranded RNA. So here's a molecule of double-stranded RNA. They bind multiple copies of PKR, and then the PKRs phosphorylate each other, and now they're active, and they can phosphorylate EIF2 alpha. So this, by the way, there's a kinase domain is KD. The DSRMB is the double-stranded RNA binding motif. That's how these molecules bind double-stranded RNA. So the double-stranded RNA allows multiple kinases to bind it, so they can be activated. They can be activated and become phosphorylated, become active kinases. Now, viruses need to antagonize that, and I'll tell you how they do that. So this PKR, by the way, is it's induced and activated by virus infection. It is induced by interferon, as we'll see later when we talk about innate immune responses. And as I said, activation of PKR leads to inhibition of translation. It leads to apoptosis and cell death. So that would effectively stop virus reproduction, except many mechanisms have evolved to inactivate this pathway. Almost every virus that we have studied has some way of inactivating the PKR pathway, because if they didn't, they wouldn't be around. It's a very powerful antiviral pathway. So here's one example in some detail. This is adenovirus. And adenovirus, when it infects a cell, produces double-stranded RNA. Now you may say, how does that happen? It's a double-stranded DNA virus, right? Well, there are transcripts made on both strands, and you can an mRNA from the top and the bottom strand, they can anneal and form a double-stranded RNA. It doesn't have any purpose, but it happens when you make mRNAs from both strands. Those double-stranded RNAs activate PKR, as you can see here. So early in infection, eIF2 alpha is phosphorylated by these activated PKRs, and you can see a gradual decline in protein synthesis. However, there is a viral RNA that is made then. It's called VARNA1, and that's a model of what it looks like there in brown. And what it does is bind one molecule of PKR. Only one molecule, no more. And that one molecule of PKR can never be activated because it needs a second PKR to activate it. It's a great strategy, right? So there's no phosphorylation of eIF2 alpha and protein synthesis remains. So this little RNA, a few hundred nucleotides long, VARNA1, prevents the shut-off of translation by preventing activation of PKR. And that is just one mechanism from one virus. As I said, viruses encode antagonists of this pathway at almost every level. And these are just some viral proteins and RNAs that counter the phosphorylation or the inactivation of eIF2. And the viruses are listed here on the right. And then the sites where the interaction occurs is listed on the left. And so we have talked about antagonists of the RNA, VARNA1 of adenovirus, which binds one molecule of kinase and prevents it from being activated. And there are other viruses that encode antagonists of PKR. There are viruses that directly mimic actually eIF2. We have a protein of vaccinia virus, which looks like eIF2. The virus makes a ton of it, and the kinase binds to it to phosphorylate it, but it doesn't, and it gets distracted from the real eIF2. There are also antagonists of other proteins that are active in phosphorylating eIF2 alpha. One of them is called PERK. This is a transmembrane kinase in the endoplasmic reticulum. Now, when it senses stress, it senses ER stress, let's say. So what is ER stress? So when viruses infect the cell, they make a lot of glycoproteins in the ER. That stresses the ER. It's too much protein synthesis. And these proteins, like PERK, get activated. They detect the additional proteins. And PERK gets activated by being phosphorylated, and then it phosphorylates eIF2 alpha. So multiple ways to counter the inactivation of eIF2. Finally, the cell responds in yet another way to virus infection by redirecting, translating mRNAs to what we call stress granules. All kinds of stress going on here in cells. The virus-infected cells, there's a lot of stress. So here we have on the top left, we have viral mRNAs that are being translated. And in a virus-infected cell, the cell has mechanisms for sensing their presence, and they shunt them into granules where their translation is inhibited. They're called granules because if you look in an infected cell, you can see punctate points of light where these are located, because you can use antibodies to the proteins that make up these granules, and you can see that they are induced after virus infection. And they contain a number of proteins, which are shown here, G3BP, TIA1, TIAR. Of course, viruses antagonize their formation. They have to, otherwise they would never reproduce. And these are some of the mechanisms that viruses have evolved to employ to do that. For example, the replication of some viruses, like hepatitis C virus, occurs on lipid droplets. We'll see what those are in a minute, but they're basically condensates of lipid. And one of the proteins that's needed is G3BP. So the virus redirects G3BP to its replication centers and takes them away from the stressed granules, essentially preventing their formation. Another example here, and there are many, you don't need to know all of them. Just know that viruses are antagonizing the formation of stress granules. Poliovirus is cleaving G3BP. So that is another component of the stress granules, so it reverses their formation. So stress granules form initially in many virus-infected cells, but the virus proteins that are then made dissolve them, allowing translation to occur. All right, next question. PKR is an interferon-induced enzyme that is activated by blank, leading to phosphorylation of blank and inhibition of translation. So for the blanks, we have GDP and EIF2 alpha. We have double-stranded RNA EIF2 alpha. We have double-stranded RNA EIF2B, single-stranded RNA EIF2 alpha, or none of the above. So fill in those blanks. All right, how do we do here? So yes, it's PKR is activated by binding to double-stranded RNA, and it phosphorylates EIF2 alpha, leading to inhibition of translation. So some of you said GDP. It's not activated by GDP. It's activated by double-stranded RNA. And you said phosphorylation of EIF2B. It's close, but it's not EIF2B that's phosphorylated. It is EIF2 alpha. And that is all on this slide here. Phosphorylation of EIF2, which then locks it into EIF2B. So it's not 2B that is phosphorylated. Let's talk about metabolism a bit. Does this cycle look familiar to you? Yeah. Did you memorize it? Yeah. Once. You never did. It was open book, right? Anyway, it's the Krebs cycle, TCA cycle, and it's, I learned this in high school also. I had no idea what the teacher was talking about. But anyway, I said, well, I'll never hear that again. And it turns out that it's central to virus-infected cells. Because when a virus infects cells, you have to make a lot of stuff to make a lot of virus particles. So you, as it says here, it places high demands on host cell biosynthetic systems. You need nucleotides, you need amino acids, you need fatty acids. And for all of that, you need energy. You need 4 ATP for one peptide bond. For one peptide bond. Now, let me tell you a factoid. I learned this last week on one of my podcasts. Your body makes, is it every day? Every day, 1 billion neutrophils per kilo of your body weight. You know what a neutrophil is? It's an antimicrobial cell, leukocyte. 1 billion per kilogram per day. It's a huge number. It comes from your bone marrow. Anyway, 4 ATP you're not impressed. All right. 4 ATP to make a single peptide bond. I think that's amazing. So it is impressive. Okay, good. Virus infection impacts cell metabolism, and this is one of the cycles, as you can see here, that's impacted. So let me tell you all about it. So here is a series of cells in culture infected with adenovirus. And the medium that we use to cover cells has phenol red dye in it, which is a pH sensor. When the pH drops, it turns yellow. And you open your incubator and you see all yellow cells. They're either infected or something else is going on. So here you can see a stack of 4 plates, mock infected at 24, 36, 48, and 72 hours. Nothing happening because they're not infected. Here, adeno wild type, you can see it's getting acidic by 48 and 72 hours. That's from the production of lactic acid, a byproduct of glycolysis, the breakdown of glucose, which leads to the production of energy. And a virus mutant that lacks one of the early proteins, E4, doesn't do this. It doesn't reproduce. On the right is simply a graph of the glucose consumption rate per million cells per hour. And you can see the wild type consumption rate goes up. So viruses need energy, and many of them utilize it in the form of glucose that is metabolized. Even rhinoviruses, you can see there, they have a sweet tooth as well. So here's the breakdown pathway for glucose. So you eat carbs. A major breakdown product of the carbs is glucose. And then the glucose is taken up into your cells by glucose transporters called gluts. It's kind of appropriate, right? If you're a glutton, the glut is going to take up your glucose. And then the glucose is metabolized all the way down to lactic acid, which makes the cells in culture yellow. But on the way, it is converted to many intermediates, as you can see here, and you get energy in the form of ATP and NADH. And down here you have pyruvate, which can be made into acetyl-CoA, which is a precursor for fatty acid synthesis. In addition to the energy that's getting made, you also have a point up here where you could take glucose 6-phosphate and shunt it off into what's called the pentose phosphate pathway, and that can lead to the production of ribose, which you need for nucleic acids. So a lot happens from this pathway. And this is perturbed in virus-infected cells in ways that I want to show you. So here we have two viruses, herpes viruses, human cytomegalovirus and herpes simplex virus type 1, which have different effects on glycolysis. And you can see here that that herpes simplex virus is inhibiting the conversion of glucose 6-phosphate to pyruvate, and it's going instead into that shunt for ribose 5-phosphate and synthesis of nucleotides. So herpes simplex virus reproduction places high demands on nucleotide production, and that's why this pathway is perturbed. Human cytomegalovirus, on the other hand, allows the pathway to go to acetyl-CoA, which is a precursor for fatty acid synthesis. And that's because that virus needs a lot of membrane lipids in order to make its particles. So we think, at least that's the hypothesis, that the membrane reproduction requirement is the reason why CMV is different. Now, herpes simplex virus also has a membrane, but apparently it doesn't need as much lipid to do that. A little bit of information on fatty acid synthesis. As I showed you earlier, the glucose degradation pathway, glycolysis, the glucose is in the cytosol. The pyruvate that is made at the end is imported into the mitochondria, where it can then participate in the Krebs cycle. But acetyl-CoA, of course, is a precursor for lipids. But if you need lipids, they're going to be made in the cytosol, and acetyl-CoA can't cross the mitochondrial membrane. So it's converted in the mitochondrial matrix to citrate, which then can be exported, then is converted back to acetyl-CoA in the cytosol. And so viruses impact this. So for example, in human cytomegalovirus infected cells, most of the citrate is pushed out of the mitochondria to allow for fatty acid synthesis. And these enzymes that are important in the synthesis of fatty acid synthase and malonyl-CoA synthase, if you inhibit them, it's acetyl-CoA carboxylase, if you inhibit them, you inhibit human cytomegalovirus production. And you see similar effects with other viruses that have membrane requirements like VSV. So this lipid synthesis, this part of the lipid synthesis pathway is perturbed. Now back to the citric acid cycle. It is also perturbed in virus-infected cells. This is, of course, a hub of carbon metabolism, and it gives you precursors for many different compounds that you have in the cell. And the input, of course, is acetyl-CoA, which is coming from glycolysis. And then you go from acetyl-CoA to all these compounds around the circle, of course. And you can see here, for example, that, as I told you before, human cytomegalovirus causes the exit of citrate from the mitochondria. This is happening in the mitochondrial matrix. And the citrate is pulled out by human cytomegalovirus infection by a mechanism that we don't know very well, so that it can make fatty acids. Now, if you did that, only that, you would eventually stop the cycle, because there would be no more compounds, there would be no citrate to run the rest of the cycle. And so what these envelope viruses do is to input glutamine into this point. They convert it to alpha-ketoglutarate, and that allows the cycle to resume. So cytomegalovirus, vaccinia virus, herpes simplex virus, they all do that to prevent halting of the TCA cycle. And herpes simplex virus pulls out oxaloacetate to be used as a precursor for synthesis of pyrimidines and purines, nucleic acid precursors. So lipids and nucleic acids can be synthesized from components of the TCA cycle. But if you do pull off components as shown here, you have to replace with glutamine. Now, lipid metabolism. So lipids are present typically as triacyl glycerols. And this is a big energy store in most organisms. You oxidize the fatty acids to get energy. You can also use them for making membranes, of course. And so this balance between oxidation and membrane synthesis is modulated in virus-infected cells. Lipids are typically stored in these lipid droplets, wherein here is a lipid at the top, a triacyl glycerol, right? And these are typically bound by lipoproteins. And when they need to be pulled into the cell, they're converted to free fatty acids and then eventually incorporated into lipid droplets. And they're stored in these droplets. The yellow part here is the lipid droplet, and then there's a protein coat on them. So this is all modulated in virus-infected cells, as you can imagine. So, for example, back to human cytomegaloviruses, which needs fatty acids to make membranes, as I told you. It actually needs an unusual kind of fatty acid, a long-chain fatty acid, where this part of the fatty acid is longer. That can vary in length, and HCMV needs longer lengths of that. To make fatty acids, you need to transcribe in the nucleus the genes for fatty acid and sterol synthesis. So they're not always turned on. And they are regulated by a transcription protein called SREBP1M. So that transcription protein needs to bind the promoter of genes that encode these fatty acid and sterol synthesis enzymes. Now that transcription factor is typically sequestered all the way up here in the ER. So there it is, SREBP1P. It's up here in the ER, so it can no longer stimulate the synthesis of fatty acid and sterol synthesis enzymes. But it's activated in cells infected by human cytomegalovirus, as shown on this slide. So HCMV encodes many glycoproteins, which are encoded in the ER, which are synthesized in the ER. That causes ER stress. And stress in the ER is monitored, as I told you before, by PERK, this transmembrane EIF2 alpha kinase. But as the PERK gets activated, not only does it lead to phosphorylation of EIF2 alpha, it also inhibits this protein INSIG1. So the red line means inhibit. So the cytomegalovirus infection is causing activation of PERK, which is inhibiting INSIG. That pulls it off of SREB1. So now SREB1 is no longer retained in the ER. So now SREB1 can move through the Golgi, eventually be removed from that other protein SCAP, and then it goes into the nuclear pore and into the nucleus. Normally, lipin-1 would inhibit the uptake of SREB-P1 into the nucleus, but HCMV is stimulating an inhibitor of lipin-1 so that the protein can get in the nucleus, turn on the synthesis of genes that are needed for fatty acid and sterol synthesis. So that's an example of how viruses can regulate the synthesis of things that they need for replication. Same thing happens in cells infected with human cytomegalovirus. This is an enveloped virus, which on top of that requires sites in the cell which have a lot of lipid in them to make its RNA and make particles. I'll show you that in a moment. So if you look at this picture here on the bottom first, this is a light micrograph of mock-infected and infected cells. These are hepatocytes infected with human hepatitis C virus. It's stained with blue, DAPI, to look at the nucleus, and it's also stained with a dye that stains for lipids. So you can see in uninfected cells, there's a baseline level of lipids in the cytoplasm. It goes up in cells infected with hepatitis C virus. So the virus directly upregulates the synthesis of lipids. How does it do that? Well, lipid synthesis is also controlled by genes that are under the control of transcription factors. And FOXO1 is a transcription factor that causes lipogenesis eventually. So typically this transcription factor would be taken out of the nucleus when lipids are not needed. Hepatitis C infection prevents its exit from the nucleus, so now you have the synthesis of lipids by fatty acid synthase. At the same time, the enzymes that oxidize fatty acids are also under the control of transcriptional activators, in this case FOXA2. So FOXA2 causes the transcription of mRNAs that encode proteins that lead to fatty acid oxidation. And hep C infection downregulates the levels of FOXA2. And if you overproduce that protein in a hep C infected cell, you inhibit virus replication because the fatty acids are being oxidized rather than used for lipid incorporation into particles. And at the same time, if you knock down the RNA for FOXO1, you will inhibit viral replication because you cannot make enough fatty acid synthase to synthesize the lipids that you need. So there has to be a balance, right, in a cell between lipogenesis and fatty acid oxidation. There is normally a balance of some kind in cells, but HCV tips the balance in favor of lipogenesis and less fatty acid oxidation. How might virus infection lead to increased levels of ATP? Stimulation of glucose uptake, increased glycolysis, increased oxidation of fatty acids, increased utilization of glutamine, or all of the above. Okay, what did we do? That's right, it's everything. They're all leading to increased levels. Stimulation of glucose uptake, which could go into the glycolytic cycle to make more energy. Increased glycolysis, same thing. Increased oxidation of fatty acid gives you energy. Increased utilization of glutamine, go into the Krebs cycle and get more energy. Finally, last thing for today, virus infection remodels cellular organelles in a big way, especially if the virus reproduction requires membranes for, say, RNA synthesis. And that's the case with poliovirus. In a poliovirus infected cell, as I showed you before, all of the membranes are dissolved and used to reform double membrane vesicles that are the sites of virus reproduction. So here, for example, on the right, we have the ER in the infected cell, and all of these membranes are repurposed to form double membrane particles on which RNA replication occurs. And so the cell becomes full of these double membrane particles. It's where RNA is made and also where the particles are made. And this happens with many, many RNA viruses. As I said, it's a way of concentrating all of the components in one place. And by the way, you see here that sometimes these vesicles will capture some mature virus particles, and they can leave the cell in a non-lytic manner, as we talked about last time. But the co-option of membranes is shown on the left here. Here is, again, a stain of polio-infected cells and uninfected cells, again with a stain for the nucleus in blue and a stain in green for lipids. You can see mock-infected cells have a certain amount of lipids in them, and polio-infected cells have more lipids. So how is this done? Well, what happens is that the lipids are stored in lipid droplets, as I indicated before. So that's what a lipid droplet looks like. You have the triglycerides lining the outside of the lipid droplet. They undergo lipolysis, and then the fatty acids can be used for more membrane synthesis, and that's the replication compartment membrane. So the infection leads to the repurposing of the triglycerides from the droplet to make these membranous compartments. And one of the viral proteins shown here links the two together. It brings the lipid droplets right near the replication compartments so that they can be easily formed. This happens in many, many virus-infected cells. And here's another example of it with hepatitis C virus, where both RNA synthesis and particle formation occurs in association with membranes. So here you have on the left the endoplasmic reticulum, and you have these membranous structures that are formed off of the ER. You can see on this reconstruction from an electron micrograph what they look like. They're spheres, of course. This is a two-dimensional representation, but they're actually spheres that are attached to the ER. And they're coated with viral proteins, including the RNA polymerase, which synthesizes RNA right on the surface of these membranes. And then later in infection, they become filled with lipid droplets, and the viral proteins are assembled around the lipid droplet, and a particle will form eventually by budding into the lumen of this vesicle. You can see one of them forming here. It's got a piece of RNA. It's got some capsid proteins there, and some membranes and some glycoproteins as well. So it's the site of RNA synthesis as well as particle formation. And this also happens for coronaviruses, that they induce the formation of double membrane vesicles in the cell, and it's on their surface. Here's an electron micrograph reconstruction of them. Again, they're spheres, of course, and the genome replication occurs right on those vesicles. And one more example I want to show you is actually a plant virus, which we don't talk about very much here, but this is a very, very interesting strategy where, again, the RNA synthesis occurs on a membranous structure. And so here on the right is the endoplasmic reticulum of the cell. And this cell is infected with tomato bushy stunt virus. The virus makes the tomato plants short, okay? They're shorter and bushy looking, so that's where the name comes from. These viruses make membranous compartments where RNA synthesis occurs, and they're closed off, as you can see here. And this is in contrast to hep C, where they're open and things can diffuse in and out, right? So this one is closed. So how do you prevent yourself from running out of energy? It's very clever. There is an interaction between a viral protein and pyruvate kinase that brings the pyruvate kinase into the vesicle so that it can make continuous amounts of RNA, of ATP, to provide the energy for viral RNA synthesis. So, and that supplies the ATP for helicases. So here is a stain for the viral protein P33, which is the viral protein that brings the pyruvate kinase into this vesicle. Here's a stain for the pyruvate kinase, which is the cellular enzyme. And then the colocalization, you can see this little membrane vesicle formation here is expanded at the bottom. It has the viral protein and the pyruvate kinase. So other genetic experiments have shown that the viral protein recruits pyruvate kinase into the vesicle so that it can supply energy for the helicase. So that completes our trip through an infected cell. Next time we'll start talking about infections of hosts.